Hello. Hello, everyone. If you can hear me, can anyone type in yes, if you can hear me, please? Oh, fantastic. Oh, hello, Jill. Hello, everyone. Oh, Anne-Marie. Hello. Nicola. Hi, everyone. It's really good to see you here. Really, really great to see everyone. Hello, Jackie. How are you? It's really not easy in this environment today, for people to want to listen to any type of contrary opinion. In fact, it's actually never easy. So I commend everyone who has joined me here. It's really wonderful to see you all. When I sent out the email, I got a couple of, or more than a couple of angry replies, and some people were really angry that I dared to express an opinion contrary to the mainstream narrative, and someone said, um, that I have joined those doctors, and am I trying to say that I should trust that we shouldn't trust the government, that the government is trying to kind of like destroy the economy? It's am I actually expressing that opinion? So I thought about it. I actually didn't send any reply back, but I thought about what she said, and I thought, yes, I don't trust the government. I'm from Nigeria, and I think my experience is really not to trust the government. I mean, I'm not going to trust the government the same way like I trust you, for example, to be human. So absolutely, I don't. And especially any kind of global government like the WHO, especially when I know that the leader of the WHO is belongs to a party that is wanted for crimes against humanity as well. So it's really not easy to be here to listen to what I have to say. And I'm going to discuss purely science. I'm going to take you through what I've been studying for quite a while now, since the pandemic was declared. And there were initially, I actually believed that something was amiss. And then, I was one of the very first people to actually buy a mask, in New Zealand. When I went to the pharmacy to get a mask, the only people there were like people from China, holiday in New Zealand. Of course, they had the news from back home. So I really believed the idea that there was a pandemic. I believed it and really got ready. But as time went on, very quickly, I searched for the science and I looked at it and I realized something was amiss. It really wasn't adding up. And as time went on, it became very obvious that the science wasn't adding up. And then over time, I've researched extensively into not just COVID, but viruses, PCR tests, and just done a lot of extensive research. And I've come to a conclusion, And so what I want to do today is to present to you a series of investigations that I've done, so you can look at the material that I have, you can look at the links that I have, the evidence that I have, and then follow it up and do your own research. So this is not about believing what I have to say, or I don't believe in this or that. I believe in the evidence. That's all I'm looking at. I believe in the logic, the reasoning behind things. So that's what I'm presenting to you today, the logic and the reasoning that I have arrived at. And so we're going to spend about an hour. I intend to spend 1 hour here. And I've prepared these slides that I'm going to go through. And I've also prepared the slides into PDF, a PDF document. So at the end of this our discussion, I will email you the PDF as well, so you can have all the evidence. I've got all the links in the PDF, and then you can do your own research. And at the end of the hour, I think what we're going to do, I will take some questions, maybe for half an hour. So at the end, we'll spend about 1.5 hours here. Okay, so let's start. I'm going to share my screen because I've got a document prepared. So I'll share my screen. And yes, so today's webinar is on why thousands of doctors, and actually there are tens of thousands of doctors today who are questioning the whole COVID-19 narrative. And I'm going to explore the science here. So we will look at the COVID-19 disease, what they're actually calling the disease. We're going to see if there's any proof of transmission, because what we are witnessing is the declaration of a pandemic. You know, the World Health Organization declared a pandemic, and then have, you know, rolled into action a series of reactions, or a series of guidelines for health professionals, for citizens to follow, because the disease is transmittable. So it's a transmittable disease, and so they've rolled out these changes in society. So we're going to look to see if there's actually any proof that there's a disease. And if there's any proof that the disease is transmitted, we'll very quickly look at how COVID-19 is treated. And then we delve into the science behind declaring a pandemic. So there were, you know, over 100 years ago, a group of scientists realized that we might come to the situation we find ourselves in today, you know, where there could be a possible microbe, a possible bacterial virus that it's playing havoc in the world. And so they laid down these series of common sense guidelines on how we can establish that there is indeed a pandemic. So we're going to look at that, the Koch postulate, and we'll look at the updated version, the refined version of the Koch postulate, the reverse criteria. And then we look at the clinical studies. So basically who relied on clinical studies, mostly released from China, you know, because when they declared the pandemic, you know, from the history, from the timeline, it originally appeared in China, you know, so obviously it would be the Chinese scientists that would carry out the studies, you know, to tell us if there was actually a pandemic or not. So we look at those studies, and then we look at the PCR test, because the PCR test is the reason why the whole world is undergoing these changes, you know. So what we hear every day is there are rising cases, you know, hundreds of thousands, millions of people being tested. So we will examine that test. We'll see how it works. You're going to go through the science. I would expose you to a lot of science, you know. So for people who maybe are not coming from a scientific background, don't worry. It's really easy. I make things easy. I've broken it down, and I've also made a copy, a PDF copy of this slide, you know, so you're going to have all the evidence. You can go through it, then you can do the searches yourself. So you don't have to believe anything I say. So when we look at the PCR test, and then we start looking at certain tools that virologists use to search for microbes. So we're going to do all that here, hopefully in 1 hour. Okay, so let's begin. So what we know is that on the 11th of March, 2020, the World Health Organization declared a pandemic, COVID-19. I'm actually going to see, let me just 1 second, please. Okay, yes. So they declared a pandemic on the 11th of March, 2020. And let's analyze the disease, you know, the COVID disease. So you know, when the pandemic was declared, as I said, I believed it. You know, there was no reason not to. It was coming from the World Health Organization, and it was actually coming from medical doctors in China as well, in Wuhan, right? They had, I'm aware of when the first rumors started to come out of Wuhan, right? So later on, when I started to come across the clinical papers that were presented as evidence for this pandemic, this is when I became incredibly suspicious. I just felt something wasn't right. And that's because these are the symptoms and the criteria for diagnosing COVID-19. So let's look at them. Over time, they've added all kinds of symptoms, but for the first 3 months, and also even till today, in WHO's website, these are the symptoms that healthcare professionals look out for to make the diagnosis of COVID-19. So in WHO's website, they says, the person must have fever, right? And they actually say the fever should be with or without increased temperature. Now, how anyone can have fever without having increased temperature is beyond me, but this is information from WHO's website. They say cough, right? You should have cough. And then they say there's got to be an abnormal chest x-ray, and the white cell count should be either low or normal. Now, this was also another really weird listing of the symptom of this COVID. Normal white cell count basically means everyone, right? You would expect, because it's called an infection, you would expect them to want to detect raised white cells instead of low blood cells or low white cells. But this is actually information from their website, low white cell count. So what was abnormal or not right about this was that these symptoms are found in countless diseases. There's no disease that literally doesn't present with fever. When your body is out of balance, fever sets in. It's actually a good mechanism, because it's to activate the body, to start to heal itself. So fever is a component of so many diseases. Cough is a component of so many diseases as well. And these symptoms are caused by a lot of microbes, a lot of bacteria, fungi, all sorts of microbes would also cause these symptoms. So this was the very first anomaly. I saw this and I thought, hang in there. Abnormal chest x-ray, anyone with chronic cough, asthma, heart disease would have abnormal chest x-ray. Cough is found in all kinds of, just like, all kinds of diseases present with cough, even diseases that you wouldn't expect, so heart disease, congestive heart failure, there's a lot of cough in that. So these were the symptoms. So what we find is, like, the so-called COVID doesn't have symptoms of its own. It's not like diseases like, whooping cough, where the sound of the cough alone, right? It has a distinctive sound, and you could say, aha, that's like a pandemic or a unique disease. No, this thing is just nothing. Look at it. These are the symptoms. And then let's look at the proof of transmission, because the reason why the world is undergoing these changes is because we are made to believe that there's a pandemic and it's transmissible. So that means we need to be careful, avoid each other so we don't get infected. And let's look at the evidence. So this is a study. I haven't included the link, but I'm going to include the link in the PDF I'll send to you. So Chinese heart patient, a young woman in Guangzhou, China, with congenital heart disease, was hospitalized for a long time. And then later on, they decided to do a COVID test. And then they found she was positive for COVID. But by then, she'd made contact with 455 people, hospital staff, other patients, her family members. So all those people were rounded up and tested, and none of them tested positive. All of them were negative. So basically, even from published studies, if there was a disease, it doesn't even seem to be transmissible. So this is another example. I think a lot of you would remember the cruise ship Diamond Princess. This was the one that actually caught my attention, before the lockdown was instituted globally. So this cruise ship was trapped, because they, I think first about 10 days when, it set off, they discovered that one of the passengers had a fever, and then when they tested this person, they found that they had COVID-19. And this is why the the cruise ship was stranded for many, many weeks. So eventually they tested everyone, and they were confined. Basically, if there's any laboratory for studying if a disease is transmissible, it's got to be this cruise ship. So you can imagine thousands of people trapped in a confined space, mingling, nowhere to go. They're right at sea. We would expect everyone to have caught the COVID. But at the end of it, when everyone was tested, what we found was like about 92% of the people in that cruise ship had no symptoms whatsoever, right? And then when they did the test, only 16.7% of them tested positive, 83.3% tested negative. So basically you had these people packed in a cruise ship, nowhere to go, mingling for weeks. And at the end of weeks, only 16.7% tested positive. So basically this is your proof. And I mean, I can give multiple examples, but if there is a COVID, then from all indications, this COVID is not transmissible. And there's been dozens of diseases like this. Okay, so let's move on. Then we look at the treatment. This is how this COVID is treated: invasive oxygenation. So rather than putting a mask, over someone who can breathe, we intubate them, and we know there are lots of risks with intubation. They're treated with high dose corticosteroids. Steroids have a lot of side effects. They depress the immune system, they thin the skin, they interfere with the hormonal system, and they break the body down, they break the connective tissue down. So these are the drugs, high dose corticosteroids. They're treated with antiviral drugs. These are very toxic medications and sedatives. So basically you have someone like, what we know about COVID is it affects the respiratory system, right? So you have people who cannot breathe, and then you're sedating them. When you sedate someone, it reduces the respiratory rate. So straight away this, and then oxygenation, there's been clinical studies done, even with going with the idea that there's actually a pandemic, and then they found that those who were treated with masks, there was a much higher survival rate when patients were treated with just a face mask, as against putting a tube down their throat, right? Because you've got to put out someone to put a tube down their throat, and then you have all these chemicals, high dose steroids. So this is what they're using to treat COVID-19. And what's actually just unbelievable is like when like in 2003 there was the COVID-1, right? That's the SARS-1. That's the first so-called problem that we had with COVID was in 2003. And then the patients that had problems were treated with the exact same treatment being used for COVID-19. And this is what a report commissioned by WHO, this is what they said: that despite an extensive literature reporting on SARS treatments, it was not possible to determine whether treatments benefited patients during the SARS outbreak. Some may have been harmful, right? So of course they will be harmful because those drugs have a lot of serious side effects. And we know intubation, these are the problems associated with intubating patients. When you intubate them, we find that up to 55% of intubations could be fatal, right? One study found that even after recovering, 58% of ventilated patients died within the next year. Some of the side effects are nerve damage and neurological deficits. And these side effects of the treatment of COVID are now called long COVID, right? So you see in the press every day, they have news about patients who say, oh, I had COVID and now I have chronic fatigue. I'm having problems hearing. I'm having problems with, feeling numb. These are all the side effects of high dose steroids antiviral drugs, these are not considered. So when you treat people with these drugs, of course, they're going to have these deficits. So that's what we're looking at here. So basically the treatment of COVID, this is a study that found in New York, 88% of ventilated patients died. In Wuhan, it was 86%, right? An article found that 66% of COVID-19 patients in the United Kingdom died. They will die because you would have to sedate someone to intubate them. When you sedate them, their respiratory rate is lower. And before someone would actually become really sick, so sick that they can hardly breathe, you can imagine the immune system is suppressed. And then when you give them the high dose steroids, of course, that's going to completely knock out the immune system. And then if they don't have a strong immune system, how are they going to fight this serious virus? This is why a lot of people ended up dead. So in other words, the therapy for COVID kills people. It's a deadly therapy. That's what we're looking at here. So let's now start our, actually the very interesting part of this webinar, which is looking at the science. Let's see what conditions should be fulfilled before a pandemic is declared. So on the 11th of March, WHO declared a pandemic, right? Let us see if WHO actually fulfilled all the conditions that should be fulfilled before you say there's a pandemic, because it's a serious thing. If you look at the global economy today, it's crippled. So many people have died. There's been like destruction of businesses, So let's see what WHO did, if they actually followed the correct procedure. Koch's postulate. So in sometime around, I think it was around 1896, not exactly sure of the date, but Robert Koch, this is a brilliant scientist. There's a German Koch's Institute. That's one of the most exclusive scientific institutes in Germany. It's named after him. So brilliant scientist. He came up with these guidelines for actually finding out if a microbe, if a bacteria is responsible for a disease. So there are certain guidelines that should be followed before we can say a bacteria is causing a disease, or maybe a virus or a fungi or microbe is causing a disease. And it took a while before this Koch's postulate was developed. I've seen certain articles, trying to demean Robert Koch, saying he's a man from the 19th century. This is just like, someone saying Newton, let's ignore, the laws of magnetism or the laws of gravity, because the people that postulated those laws lived hundreds of years ago. So this is what these kinds of articles are trying to do. They're trying to say, we live in a modern time, we cannot apply these rules. But let's go through Koch's postulate. It was a very logical, really reasonable order, guideline for actually, finding out if a microbe is responsible for a disease. And it took a while before he developed the postulates with Jacob Henle and Frederick Loeffler. So let's see what the postulates are. So these are the 4 postulates. Number 1, it says, the microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms, right? So it says, if before we declare a pandemic, we've got to know that the virus or the bacteria or whatever is causing it should only be found in people that are sick and not those that are healthy. It makes sense. If you're healthy, you're healthy. Right? We only worry when you're sick. So when you look at these postulates, please also think about what's going on today, where, while our lives are being destroyed because healthy people are testing positive for COVID, and they're talking about cases, healthy people testing positive. So that's the first postulate. The second one is, the microbe must be isolated. This is the word you've got to note. Isolated from a disease organism and grown in a culture. Isolated. You've got to extract the organism from wherever you're looking at, maybe in the blood, someone's blood or sputum or whatever. You've got to extract the microbe, culture it. So you put it in a put the sample in a petri dish so that we would know that there's a specific microbe that we're talking about here. The third law is a postulate is that the cultured microbe should cause disease when introduced into a healthy organism. So you've extracted or you've isolated the microbe. Now if you introduce that, so you have this virus or this bacteria. If you introduce it into an animal, for example, the animal should develop the disease. And then when the animal develops the disease, the fourth postulate is that the microbe must be re-isolated from the inoculated disease. So you've got to get a sample from the animal that's developed the disease that was healthy previously, and then identify the bacteria. Then you can now say that you have a bacteria that is actually causing a disease. So I give a lot of credit to Robert Koch, because even though he formulated this postulate, he wasn't able to satisfy them. So he tried to prove that, I think it was either cholera caused diseases, and he wasn't. So it's very interesting that you can see that the postulates are very logical, that he was able to come up with them, even though it actually didn't progress his career in many ways. So now what happened was when viruses was discovered the viruses were were not discovered until electron microscopes were discovered in the 1936. But a lot of scientists speculated that there might be organisms that are smaller than bacteria. So when electron microscope was discovered and people looked at samples, and then they found microorganisms that were even smaller than bacteria that seemed unable to survive outside the cell. What they say about viruses is that they don't survive outside the cell. So another brilliant scientist, Thomas Rivers, modified Koch's postulate, because Koch said that the microbe should be cultured, but viruses cannot survive outside living cells. So this is what the Rivers criteria says, that the first one is that the virus must be isolated from a disease host. So if you put it, side by side to Koch's postulate, you would see it's more or less very similar. So you have to be able to isolate the virus from a disease host. You must cultivate the virus in the host cells. So because the virus cannot survive in a petri dish, you've got to be able to show that it's actually inside the cell, right? You've got to offer proof of filterability. So now, since this virus can only survive inside the human cell, you have to be able to filter out all the other particles like bacteria, human cells, anything else that isn't human cell should be filtered. So we know anything else that isn't, the virus in a human cell should be filtered. So we know we most likely have the virus. And then you should be able to produce the same disease in a host. You should re-isolate the virus. So if you introduce the virus into an animal, the animal should get the disease. You should be able to collect a sample from the animal and then re-isolate the virus. And then you know that you actually have a unique virus that is able to cause disease. So these are the scientific criteria. These are the guidelines for establishing a pandemic. There has been modifications since then, but those modifications have never been widely accepted by mainstream science because they don't make as logical sense as the Koch's postulate and Rivers criteria. So far till today, this is what is being used to actually prove that a bacteria, a virus, any microbe it's associated with the disease that scientists or anyone is associating them with. So let's see what WHO did. Did the World Health Organization isolate the COVID-19 virus? So let's have a look and see what WHO says about this. So basically, WHO says, yes, they cultured the virus, right? And then they also say here, and this is from WHO's website, that the virus, it says the Chinese authorities identified a new type of coronavirus, which was isolated on 7 January 2020. So WHO is actually telling us that the virus was isolated. Now, I'm looking, I've highlighted isolated, and I'm stressing on this word because you would find out very soon that this is a lie, that the virus was never isolated. And WHO, the World Health Organization, has on its website a claim that the virus was isolated. All right, so let's look at these studies that WHO relied on to declare the pandemic. There's about 6 or 7 of them. There's actually 4 main studies, and they're all from China. You know, the pandemic was declared in March. I think the virus was noticed in China sometimes in January. So by February, March, scientists had actually published studies and done a lot of research and then confirmed, according to them, that we had a unique virus that was causing a unique disease. So we're going to look at the published studies, you know, but what happened was these are journalists, Torsten Engelbrecht and Konstantin Demeter. These are Swedish journalists, and they wrote to these scientists, you know, to actually verify from them if their studies, you know, if they're claiming in their studies, because I've read the studies and there's nowhere in the study that shows that the virus was isolated. Absolutely not. So what these journalists did was they wrote to them. These are journalists from the offguard.com, off-guardian.com. That's their website. They wrote to the 4 groups of Chinese scientists to ask them if they isolated the virus. And let's look at what they said. Now, these are the studies. I'm going to send you this PDF. And this is one of the studies. This was published in March 2020. These are the studies that WHO used to declare the pandemic. They relied on, these are the studies that backed the declaration of the pandemic. So the scientists said here, you know, because if you look at the study, this particular study, there's an image of something they call a coronavirus. You know, I'm going to publish a link to a beautiful article that I would encourage everyone to read. It's an amazing piece of work. So that article shows an image, this image that they're calling coronavirus, and also shows images of other structures in the human body that resemble this thing that they're calling coronavirus, right? So this is the reply of these scientists. They said the image is the virus budding from an infected cell. It is not purified virus. So what you would realize from these replies were like these scientists, you know, they were not trying to be straight, right? Because we've read the studies, I've attached the links. You can read the studies yourself and see where they actually specify that the virus was isolated. They never did. Okay, so these particular group of these scientists were asked, and this is their reply, that we could not estimate the degree of purification because we do not purify and concentrate the virus cultured in cells. Okay, this one said we did not obtain an electron micrograph showing the degree of purification. And then this study in February said that we show an image of sedimented virus particles, not purified ones. So none of these scientists verified what the statement WHO made that the virus was isolated. So basically, this is a lie. This is a lie. There's no isolation of any virus. There's a reason why you would see later on why I'm emphasizing on the fact that the virus was not isolated, whether as a singular organism, which is impossible, or even as a form in which microbiologists recognize it. So they recognize the virus only exists inside a human cell. That was not done. Okay, we're going to see that later on. All right, so I want to analyze this study. I've picked up a study. This is basically more or less how these, when you look at these studies, that's what you would see. They have an arrangement for clinical studies. See, like there's a lot of language in these studies that are meant to discourage you from reading them. You would think, oh, who am I to understand these words? But when you look at any study, there's always a conclusion, right? Just look at the first paragraphs. That's where most of the information is. The rest is just basically padding. So let's see how this is a study in Australia that said that they have identified the COVID-19 virus in the first patient in Australia, right? And then this is their method. This is, I've just copied the study. It says here, real-time reverse transcription RT polymerase chain reaction testing for SARS-CoV-2 was performed on material from an initial nasopharyngeal swab. So basically, what I'm trying to point out here is there's no purification. Remember how the emphasis is on isolating the virus. So in other words, the reverse criteria said you have to show filterability. So you've got to show that you're able to filter out the sample you're using. So these people are using the nasopharyngeal, that swab from the throat. Before they put it into the PCR machine to do the test on it, they should have filtered it. And the reason for the filtration is, since they're suspecting it's a virus, they should filter out the bacteria. I think a lot of you who've come for the scan, you know how the human body is filled with microbes. I think 30% of the scans we do, or 40% of the scans that we do, we know people have multiple microbes, all kinds of viruses and bacteria and other microbes, right? So this is why you need to filter any sample, especially if you're trying to prove a unique virus. Of course, you have to filter the sample, filter out the bacteria, and so you only have human cells that have viruses. This was not done. What they did is they just took the sample from the throat straight into the PCR machine, and that's actually how they're testing for COVID-19. And you can see here again that says materials from the initial nasopharyngeal swab was used to inoculate a Vero cell line. So basically they got this gunk from the patient's throat and straight injected it into the cell. So what they injected, of course, has other particles in it, right? It's not just a virus in human cells. It's got bacteria, it's got all sorts. So this is, and I mean, this is the format for all the studies. All the Chinese studies was exactly this that they did. So when you're looking at these studies, what you're looking out for is to see if the studies were filtered, if there's any filtration done before the material was used in a PCR test. Okay, all right. So let's look at how to purify a sample, because I was really interested in finding out how this is done, because I couldn't understand why these scientists were not just doing this supposedly simple process. And it is a simple process. A lot of companies do this, companies that manufacture lots of drugs, or even vaccine companies, they have to filter out whatever solution they're using, so they know that they only have like very tiny particles. So this is how to filter and isolate a virus. You collect a sample that could either be from the throat or blood or urine sample, you run it through a filter, and what you're doing is you're passing it through a filter that is able to hold larger particles. So all the bacteria, human cells, and fungi, these are much bigger than a virus, right? They stay behind on top, and then whatever is lighter, or you can even just hold back the bacteria and filter out the human cells or whatever cells you're using, since they contain the virus. But either way, you've got to run it through a filter. It makes sense. You have to filter what you don't need, because the PCR test, as you'll find out, is incredibly sensitive. So run it through a filter, and then you pass the filtrate through a density gradient solution. So basically, once you passed it through a filter, they filtrate, you now add this liquid to it. And what that liquid does is when you spin the liquid, then the viruses would now collect at different bands, right, in the solution. And then you can pipette the cells in with the viruses, and then that's what you use to do your test. And like I said, this is, you can just like type in how to purify or filtration, viral filtration in your Google search, and you would see dozens, literally first page, dozens of companies that do this process. But the scientists that actually performed the studies that led to the declaration of the pandemic, for some reason, couldn't do this simple procedure that's absolutely required before you could say you have a new virus. All right. So this is more or less, I've just gone through what should be done, which the scientists that published the studies didn't do. So let's look at the PCR tests. The PCR test is the test that is used to find out if someone has COVID-19. Let's see how this test works. So the PCR test is actually a test for DNA, deoxyribonucleic acid, right? It doesn't test for viruses. DNA is they call it the building block of life. And this is what DNA looks like. It's like a helix, like it's twisted like a ladder. And then here you have these base pairs. So the DNA is made up of just 4 proteins. I think there's the DNA and then the RNA. RNA is found in viruses. So for you to detect viruses with the DNA, with the PCR test, you would have to convert the RNA inside the viruses to DNA. So, but that's what we will be testing for with the PCR machine. We look for these strands that have these base pairs, and these base pairs are complementary. So adenine always pairs, they're paired together, and adenine would always pair with thymine, and cytosine would always pair with guanine. So you have A pairing with T and C pairing with G. It's always like that. And so this is kind of what a sequence, a genomic sequence would look like. You would have the main strand, and then you have a complementary strand. So A is always with T. If you see T here, you would see A. If you see G, you would see C, right? And then it carries on. What the PCR does is it tests for little bits of DNA, right? It can only test for a maximum of about these are sequences, these are base pairs, and they're also genetic sequences. So COVID-19, they claim it has about 29,000 of this. So these numbers like this, ATTA, it carries on 29,000 times. And it's always changing. So maybe the next, if this was the genetic sequence for COVID, the next sequence here might be like another TA or AT, and it carries on 29,000 times. But the PCR test cannot test for the whole sequence. There's no way it can test for 29,000. It only tests for bits of 200, depending on the machine, but usually just a few thousand, and that's the maximum, and so that's what it tests for. It doesn't test for viruses. You would have to, if you introduce a sample into the PCR test, you would have to, the PCR breaks it down, into little bits. And then those little bits is what you're testing for. So the PCR, the viruses do not contain DNA, and I've made a little note of, what the DNA is, okay? The PCR is a DNA manufacturing technology. It amplifies or replicates DNA. So what it does is if you introduce something that contains DNA, and every living thing contains DNA. So maybe let's say this is our filtered sample, and then you introduce it into the PCR machine, the PCR would now amplify it. So the way it works is, let's say you have a little bit of something, the strand. Remember, this is the structure, right? It always has, the DNA always has a main strand and it has a complementary strand, right? So when you introduce it, the PCR machine would break this strand into two. So you have two separate bits, right? So you have the main strand, and then you have the complementary strand. And it does this by heating up and cooling the sample rapidly. By doing so in cycles, it just breaks it down, okay? And when you're using the PCR, you would have, because the PCR test cannot detect anything from scratch, you've got to tell it what to look for. You cannot introduce a sample into a PCR test and expect it to tell you what it is. You would have to introduce a little bit of what you think is in the sample before the PCR test can now replicate. So when you introduce that little bit of what you think it is, so what happens is the PCR will now break it, break that the material that the sample, the DNA in the sample, break it into two, and then start to copy the bit that you've introduced. The other thing you introduce into the PCR are these bases. So you're going to put solution that contains this adenine, thymine, cytosine, and guanine. So when you break this into two, then the one side of it would now find the matching parts, and then the other complementary strand would also find the matching strand, and then you have two, right? Then the process repeats itself. You break the two strands, and you have 4. And then from 4, you have 8, you have 16, and like that. So if you do that, 10 times, you have 1,000 strands. I think if you do that 1,000 times, you would have up to 1,000,000 times whatever you started with. And this is what the PCR is used for. So if you imagine in a crime scene, right, and someone commits a crime and murders someone, or and cleans up the place very well, and there's no sign of anything that's happened there, and maybe like a scientist comes along and suspects that a crime took place in a particular position, and then they take a little bit of sample, just rub cotton board on the floor, and then maybe they can't even see what they are looking at, right? And then they take that to a forensic lab. So what the forensic scientists would do would be to introduce that sample, filter it, and then introduce it into the PCR machine, and then add a little bit of the suspect's genetic sequence, because they want to read to see if the DNA belongs to the suspect. And then the PCR machine would now start to replicate, because the sample is too small to do anything with. The PCR would magnify it 1,000,000 times. And when it's magnified it 1,000,000 times, then it becomes possible to match that little bit that was collected to the victim's DNA profile. And then that's how we can tell if the blood actually belonged to the victim or not. So PCR has its, a lot of benefits in forensics, and that's usually where it's used. It's hardly used. It's never used for any kind of test on a large scale, apart from what we're seeing today. So that's the function of the PCR. It is not a device for testing viruses. It replicates little bits of DNA material. It replicates them like 1,000,000 times, so that you can now compare what you've replicated with a database of DNA, and you can say, aha, okay, this will now know that we have enough to know that this DNA belongs to this or belongs to that. And that's how it works. So the PCR is a cutting-edge technology for replicating DNA. It does not test for viruses. And on many occasions in video, Dr. Kary Mullis, this is the man who invented, brilliant scientist who invented the PCR, said, I'm also going to add links to the videos, where he's said so many times that the PCR test does not test for viruses. Unfortunately, Kary Mullis died August 2009, just a few months before the pandemic was declared. Okay. So to use the PCR to identify a virus, you must add a bit of DNA that you expect to match the virus. This bit of DNA is called a primer. So the PCR does not detect anything from scratch. You've got to tell it what it's looking for. And that's the one of the biggest problems with the studies, which there's no time to analyze those Chinese studies properly. So it's like, how do you know you have something new when you don't know what you're looking for, right? And it's a whole new discussion where the scientists are talking about de novo assembly, right? Where they now, what they do is, because they don't, like they say they have a new virus and they don't have a little bit of that virus to add into the PCR to determine if they actually have a virus. So what they do is they take bits of the DNA that they have discovered, they run it through a software. The software just makes them up, puts them together. And this is how they arrive at a new sequence, because it's impossible for them to have actually determined that a new virus existed with a PCR test, which they're claiming, because they would never have a little bit of that new thing. They don't know what they're looking for in the first place. So this is the problem with anything concerning the PCR. You need to know what you're looking for. It is not used to identify a new microbe. That is impossible, because it needs a little bit of what you're looking for to work. Okay. PCR cannot detect DNA from scratch. So moving on, this is what happens. This is how the PCR actually it's used, so to do the test. So you add the filtered sample. So you've got to filter the sample. So you only have viruses, for example, or you only have bacteria. So you add that into the PCR, and then you add your probes and primers. The primers are the little bit of known DNA you need to add to the mixture, because for the PCR to work, you've got to tell it what to search for. So somehow you've got to have an idea of what you're looking for before it works. So you add the primers, which are little bits of DNA. The probes are like colors. The way the PCR works is, once it's identified the little bit that you've added to it, it changes color. And that color tells you, aha, we've nabbed it. What we added, what we suspected is what it is. And then you turn on the PCR, you cycle the PCR. So the cycling is heating and cooling, and this is done up to 40, 45 times, usually much lower than that. And then to magnify the DNA in the sample, you watch for the color change, and that indicates a DNA match. So basically, this is how the PCR test works. You need a probe, you need primers, you need to filter the sample, and then you do your cycling. If you do not filter the sample used for the PCR test, you know what would happen. It may contain the DNA from viruses, bacteria, fungi, and human cells. So if it's not filtered, then you have DNA from everything, right? This is why it has to be filtered. The Wuhan scientists did not filter the samples used in their studies. I have a list of all the studies. All you have to do is just to go read the studies for yourself. Don't believe anything I'm saying. Read the studies and find out for yourself. And I'm going to send this PDF to you. So the COVID-19 DNA has gene sequences from bacteria, viruses, fungi, and human DNA. This is what we're going to verify live in the next few minutes. You're going to see, this is the COVID-19 sequence. There's about, I think, 29,000 of these blocks of code. This is code. And that's what is published in WHO's website. And it's called the sequence of COVID-19. But in this sequence, we will demonstrate live here. You will see that this sequence has codes from known bacteria, viruses, fungi, and even DNA. You will see the consequences of this later on. So we have the microorganism that is being tested has little bits from the human body. That's what we're seeing here. Okay. According to WHO, COVID-19 has 4 specific gene sequences. So they've done all their studies around the COVID-19 virus. And what they came up with is there are 4 genes that are specific. So once you see those codes, a code, remember, the sequence is a little bit of DNA. Nobody, no test can do the whole virus. 29,000, that's impossible. You can only do little bits, 150 here, 500 here. This is what you do. So they've said in the 29,000 sequence of the COVID-19 virus, there are 4 sequences, maybe about 50 genes, not very much. But once you see them, you have COVID-19. And these sequences is what is now used in the kits. They have all these test kits, all this testing. They don't test for the virus, the whole 29,000. That's impossible. You've got to test for a little bit. And this is what they're testing for. So they call these sequences, these codes, RNA polymerase gene, N gene, S gene, E gene. This is what they call them. And they are codes like the CT, GATT thing that we've looked about, this code here, right? So there are some of them in here that are specific. The minute you see them, you're dealing with COVID-19. And these are the sequences that are searched for with the PCR tests, right? So to detect COVID-19, you've got to add the sequences of these genes. This is the primer. So the primer has got to match these specific genes. When you add the primer and the minute it changes color, okay, there's N gene, this is COVID, or S gene, that's COVID, or E gene, that's COVID, or RNA polymerase gene, this is how it works, okay? So 4 of them, 4 specific gene sequences. But here is where it really starts to get incredibly troubling. I mean, this discovery deeply troubled me, still is. Like I said, you are going to do this test yourself. We're all going to become virologists here at the end of this. So what we find is like these 4 specific gene sequences that are used to identify COVID-19 are found in many bacteria and human cells. They are found in many bacteria and human cells. In fact, many of the genetic sequences of COVID-19, the 29,000 complete gene sequence of this microbe that they say is unique, that the sequences are found in dozens of other microbes and human cells. So basically, when they're doing the tests and they're testing for these genes, they are testing for human cells. They are testing for Staphylococcus bacteria, Campylobacter, all kinds of bacteria and viruses that share the same sequence. So there is absolutely nothing unique about COVID-19. When COVID-19 is being tested for, they're testing for when they put the swab down your throat, they're scraping some of your cells. Of course they will, right? They're collecting samples. Maybe you had a flu, or maybe you had a cough, that cough caused by a bacteria. They're also collecting those samples. And that's what they're going to test for in the PCR. And then they say people are COVID positive. We are going to demonstrate this here. That's actually the main thing. We're coming to the end of this webinar. I think I said, yes, I've got 3 minutes to go. So like I said, the primer, this is the bits that you have to know to introduce into the PCR tool, because you've got to know what you want to look for before you use the PCR. So that bit, the primer, used to identify COVID-19 is found in many other microbes and human beings. And this is the code. I'll send you this. We're going to look at the COVID-19 code. Then we're going to look at the primers and probes approved by WHO. And now we're going to use the technology that virologists use to search for gene sequences. We're going to search for the probes and the primers that are approved. These are the things that are tested for. So we're going to search for these probes and see whether they match human cells or they match other microbes. We will do that with this website, and I've told you how to do that, and we're going to do it here as well. The COVID-19 PCR test is testing for DNA from human cells and other microbes. Do the search yourself. Do the search and see for yourself. Do you still believe in COVID-19? So that's what we're going to do next. I'm going to minimize this program, and then I will just open my browser, and then let's do the search. So this is a public, this is called GenBank. So this is where you go to see the genetic sequence of anything, any microbe, human beings, viruses, bacteria. You just type in the name, and then you're going to find a microbe. And this is the genetic sequence of COVID-19. It says so here, sorry about this, severe acute respiratory syndrome, coronavirus 2 isolate, Wuhan 1 complete genome. And that's the sequence. That's where it starts. And this is the sequence. This is how they list it, from the very first sequence, and then it keeps going on, 1, and then you have the ends here 60, it starts here 61, and carries on. And we can see here, we have 29,881 or something like that. So that's the sequence. So it's from this sequence that the World Health Organization, this is their website, this is World Health Organization, they have recommended probes and primers. They've got these from this sequence. They've got it from here, somewhere here, they got these. And they recommended, they say, this is what we have to test for. This is a primer. They have all different recommendations of what we need to do. These are the genes. This is for China, what they're testing for, the RNA polymerase and the N gene, these 2 targets in RNA. So they all have little bits of the specific gene. Each country is testing for something. So this is a primer. Just let's copy it. So what we're going to do now is to look at this, because this primer, like I said, is copied from here, somewhere here, right? And then what we'll do next is to go to the BLAST public domain. This is where you go to compare any sequence at all. You want to see if that sequence is found in anything else. This is where it's done. These are public domain sites. It's called BLAST, right? So I would, first of all, what I'm going to do is I'm going to see if that sequence is found in human. So I'll click on human, and then we can go to microbes later on. Okay, and now I would enter, just paste that sequence. And then what I'll do now is I'll make sure on that program selection, this program is going to show me the highly similar sequences, right? Make sure this is checked. Click on BLAST. And it's running the search. It takes maybe about a minute or so, depending on the length of the sequence. So this is the first one we're going to do. And we're going to see the result it's going to give us. So I encourage you, I will send the PDF with this link. I encourage you to actually, what I've been doing is just copying codes from here and running the search. This is very, very disturbing. So why does this have human genetic sequences from bacteria? The reason is those Chinese scientists, whether by, I don't believe it's carelessness. It's most likely criminal, right? They didn't filter the samples that they used to determine the code, the so-called code of COVID-19. So because they didn't filter the sample, they put it inside the PCR machine, and then the PCR, and then they started testing for sequences from all sorts, right? And of course, they were testing for sequences from human cells, from other kinds of bacteria, from all sorts in the samples. And because they also didn't have the original COVID-19, they didn't know what they were looking for. So they used a system called de novo assembly. So basically they would now tell a software, this is the realm of artificial, just nonsense, not real. They would put this something, a code inside this, one of the codes that they've got from the PCR, they put it inside this software, they run the software, and then the software would now kind of like put the little short links together end to end to build this thing, right? And then of course it's going to build with all the codes in there, all the bacteria, viruses, human cells, and then that's why you have 29,000 base pairs, and it's all full of sequences from all kinds of other organisms. So let's look at our search and see what we've found. Now we've done our search, it's completed, and this is what we are finding, that the primer, what it's been tested for. So when they take a sample for you and from you, and maybe that lab is using this primer, it could be any of them. We're going to do a few tests here. It's using this. So this is what they would use. They would tell the PCR machine to look for this inside your sample. But this is what we're finding here, that the primer, I don't know how to get rid of this now. Right. It contains human cells. Human cells. This is it. Human. 100% identity match. All kinds of chromosomes in humans. This is what we're looking for. This is it. So when they do the test, they're actually testing for chromosomes from your body. This is, and then when they find something from your body, they tell you you are positive. That's what they're doing, right? Let us copy another one and now check for microbes. Just like basically we'll copy them at random. I haven't done these. I've done quite a few searches here. I haven't done a lot. Now let's look at microbes. So you have your cough, normal cough, you have fever, which lots of bacteria, fungi would cause. You go to the hospital, they suspect you of COVID, they put inside the PCR this primer, and then let's see what we're going to get. So let's see if there are viruses or bacteria or microbes or other things that would cause fever, would cause cough, would cause sickness, that share the same sequence as what is being used to test your body for COVID-19. We're running the search here. You know, I encourage everyone to do this search. I have, you know, I kind of discovered this a few days ago. I've known about the PCR, but I'm actually taking the time to do the search myself. You know, I think many weeks ago, there was this woman that first alerted the world that some of the sequences that were in the coronavirus were you know also found in human beings. You know, so it's kind of taken us quite a while to really start to look at things properly. You know, to look at the sequence, to now do the search ourselves to find out what's going on. So we've completed that search, and this is what we are finding. We are finding that that sequence, right, that they're using to test for COVID from the swab sample, these are all the microbes that share the same. Like this is probiotic, lactobacillus, right? These are bacillus. These are microbes. Some lot of them I don't know of, but they're there. Most of them probably wouldn't be able to cause diseases. Mycoplasma. Some of them you recognize. E. coli, right? E. coli. So you have dysentery, right? They do the test. It's going to come up positive. Look at it. Probiotic. Maybe you take a little bit of probiotic that morning and have a little bit of fever because your body's trying to heal itself. You go for the test. That's it. You're positive. These are all the bacteria that share, and these ones are incredibly high query rates, 89% match, absolutely high matches, right? Very, very similar. This is what they're picking up. So, well, that's actually what I really wanted to introduce everyone to the science, you know, because the media is totally blacked out any kind of discussion around COVID-19, any kind of intelligent discussion. Nothing is encouraged anymore. And it's got to a point where we all have to become scientists, because you see, what is going to happen is they are basing their vaccine on this code, right? So they are building something that is anti this code, anti codes inside your body. They're going to inject, they're producing vaccines that contain the opposite. That's what a vaccine is. A vaccine would want to destroy the virus, right? So it's got to destroy these codes. And a lot of these codes are your human genetic code. So the vaccine, it's impossible that the vaccine will not harm the human body. But I think it's even going to do a lot worse than harm the human body. The vaccine is going to transform. This is a time that I never suspected, that such a thing could happen. I knew that things were not right. Medicine. That's why I practice natural medicine. There's all sorts of awful stuff that goes on in mainstream medicine. And I realized this very early on and decided I was only going to do the right thing. But to imagine that the corruption will be taken to this level, it's unbelievable, right? Where they are banking on everyone being too stupid to think logically, right? They have introduced a disease that doesn't exist, a test that isn't even used for what it's not even meant for that purpose, the PCR. And even if you want to imagine it's meant for that purpose, it's actually testing for human cells and other kinds of microbes, many of them harmless in the human body. And they've declared a pandemic and they've instituted this change. I mean, look at what has happened to humanity since the pandemic was declared. We've gone into global lockdown. They have banned people from socializing, right? Their so-called guidelines, what they're advising us to do are steps that would further isolate humanity. They're pushing us away from each other. And we know that isolation, there's actually a study, I'm going to find that and attach it, that was done many years ago. Very, very extensive study. And they were looking at the factors that put the most pressure on elderly people, factors that would literally, yes, have the capacity to cause the most disease in a person. And they found it was isolation. When you isolate human beings, we die. We are social animals. And then you find these measures are measures that are telling us that we are bags of diseases. We should cover our faces. That your face is like the human baby up until I think 18 months or even a little bit older than that. The most interesting thing in the universe for a human baby is the human face. They don't care about anything. So we don't even know the effect of masking ourselves up, covering our facial cues. We don't know what the effect is on human psychology. These are what they've introduced based on a lie, right? Look at the language, lockdown, what are all the social distancing, right? These are all masking, introduction of mask. These are all languages. And if you look at what they're doing as well, so everything they're advertising is basically what is destroying our source of joy, so they've banned restaurants, so we cannot meet and socialize anymore. Theaters are closed. Sports, music, these things are all banned. Even just speaking to people, they are banning. They say singing, for example, encourages the spread of a virus, something that doesn't exist. And they are moving on with these steps. They are talking about a reset, that they're going to reset the global economy, and we're seeing it happen, right? And everything is happening. They've destroyed the global economy, right, based on this, and destroyed businesses, and everything is based on a lie. On a lie, You can speak to, what I encourage everyone to do is to understand the science. I will be putting out information out there. I'm going to put everything inside a particular page, so when you go to that page, you would find other links. We need to become scientists, so we can educate other people. It's got to that point. That's the reason why they're doing this. Is they and when I say they is whoever is in control. There are people who, for example, they are the they. They are carrying out this lie, because this is a lie. By the time you do this search, you realize something is not right. Perhaps I might be wrong. I don't know exactly what's going on, but what I know is something isn't right. The science is not adding up. Well, that's really all I have to say, people. I hope, okay, I can see. I hope you can make sense of this discussion, and then it means something to you. I don't know. It's still mind-boggling for me. I'm still digesting, what I've been discovering since the past months. So if anyone has any question, I think it's time to just take a few questions, and then maybe we'll round up in about 15, 20 minutes. I think what's important is, I'm going to put this out, send this to, the document to everyone, so you can do the search yourself. You can research out, send a number of links. You can read the studies. You can ask questions. If you have doctors or people that are just like unable to comprehend that such a thing is possible, introduce them to the science. It's all about the science here. All right, so anyone with any question, I'll be really glad to take any questions that anyone has. Let's see. An isolated, absolutely. A PCR test cannot diagnose Kary Mullis, the PCR inventor, yeah? Stated this. Everyone, I think a lot of people here have already know what I'm talking about, which is good. So sorry, I have to leave now. Okay. So let's see if there's any questions. So what is this disease that is affecting so many? Right. So basically, like I've looked at death rates, so there was one I saw yesterday. I think it was someone comparing death rates in England for October, and they found it was exactly the same. There was just only 7 more people that died or so. So I think what's going on, as you can see, is every disease is now a COVID disease. So when you look at it, at the end of the day, we are going to look at the figures. We would find, at least from March, April, May, June, July, up until September, you were going to find that the death rate in many places remained exactly the same. So what you're going to notice is any disease, there was lots of reports of people who were hit by a bus and then they died of COVID. So the COVID is, any disease is now called COVID. That's what I think is going on, right? This is what the so-called, that it's affecting a lot of people. It isn't. People are sick. People have always been sick. And also, it's very difficult to put the scale of death. We do die. We're not built to live here forever. It's hard for people to imagine that every year 50 million people die worldwide, right? So if they say so far 1 million people have died from the test, we know they're dying of so many things. Even if you put that into globally, if you put that into perspective, it's just doesn't add up as well. So I don't think there's any that there's a disease occurring, but that's a possibility. That's a possibility. It's very possible that people are developing. There's a lot of evidence which we could look at some other time, which is suggesting that the frequency, the harmful electromagnetic frequencies, we're looking at 5G now, for example. There's a lot of evidence, suggesting that these places like Wuhan, we know Wuhan launched 10,000 5G substations just a few months before the pandemic, right? And we know that interferes, that the frequency of 5G at around 30 gigahertz, or is it 60? I'm not exactly sure, but it interferes with respiration. So it is possible that's what we're looking at. But whether people are actually dying en masse, this is not our experience. You know, I have family everywhere in England and Nigeria. You know, I'm in New Zealand. Okay, maybe nothing is happening here. I have friends everywhere. No one has that sense of people just dying. It's just not happening. You know, it's only happening in the media. Thank you for the explanation in layman's terms. Will you upload this onto YouTube or BitChute? I think I will. I probably will upload it. I'm not into YouTube, but I'm going to upload it into YouTube. Okay, so besides educating as many as possible, what else do you think we can do? I don't know. This is beyond me. The scale. I think it was one of those Nazi, you know, what you want to call them, Nazis, that said, you know, the bigger the lie, something like that, the more difficult it is for people to believe it's a lie. The scale of this thing is unimaginable. But I think nothing beats facts, right? And I think the weakest point for the lockdown is the PCR test. The PCR test is the test that we can prove legally that it's absolutely wrong. And it's the governments are not. If you, I saw something I was going to add to this webinar, where if you go to the Australian government website, they're saying the PCR test is not reliable, right? So, and that's like across the board. So that is something we need to understand. We all need to understand the PCR tests, and I've got a series of brochures that I've written. We need to understand that properly. And then we need to understand this sequencing. We have to speak the language. We can't be afraid of science. These are words. So a lot of it is, even for me, they use like language, a particular language, to discourage us from reading the literature. You should learn to skip things that don't make sense and just focus on the things that you can understand. Even in those things, you're going to see a lot of loopholes. So we have to start to speak this language and explain things to people. Talk about genetic sequencing, talk about the codes, talk about using blasting these genes, how they're matching human genes, how they're matching other microbes, pointing people to these websites to carry out the search themselves. I think that's one of the best things we can do. Can you see an end to all this? I hope so. I really hope so. It is frightening. It is frightening. I hope so. This thing is based on a lie, so it's deliberate. It's deliberate. The people behind it know absolutely what they're doing. It is not ignorance. Perhaps a lot of us many doctors are ignorant. A lot of politicians are ignorant. That's true. But there are the masterminds behind this. Someone like Fauci knows what's going on. He has to. He knows what's going on. Maybe a lot of people are ignorant. A lot of heads of states are ignorant. I believe it. But a lot of them would also know that this is not right. This so-called second wave is based on cases, positive PCR cases. The deaths are similar to a normal flu season. Absolutely. There are so many German lawyers tackling governments. Absolutely. I know about the German. This is also what we're going to pursue, to see if we can overturn the lockdown, if we can expose the PCR legally. This is something to try. Absolutely. I agree. So fear causes stress, and stress reduces your immune system. So that's the worry. See, I worry that we are going to start to experience the high death rates in the months to come because of the stress, because of the focus on COVID. And then, so imagine someone with a serious problem goes in, has fever, has cough, has an enlarged X-ray, could be congestive heart disease. Straight away, they test the person, find out they're COVID positive, put them on these drugs. Those corticosteroids literally would just destroy anyone with like any kind of congestive heart disease, right? And then that will kill you off. So the consequence, I think, as time goes on, we might start to see the high death rate that is coming from stress. As you pointed out, stress on our systems and literally starts to kill people off. This is what I think could happen if this continues. I agree with you. There are serious evil masterminds. Thank you so much for this. Well, thank you, everyone. I'm really glad that, you know, you turned out, you know, I'm glad that we still have people who are logical thinkers, people who are free, you know, to think. That's all you know, that's kind of what I wrote in the email. You know, it's like, listen, you know, if you don't have an open mind, it's the same way I'm going to go to listen to anyone who says, I've got the evidence that there's COVID-19. I am going to go with an open mind to listen, because I really wish I was wrong. But there's no evidence. There's not a single evidence supporting this. Not a single evidence, not from the published studies, not from the government websites, not from the people pushing this, not from the newspapers, nothing, zero, zilch, you know. So I admire everyone, and I thank you all for coming. And I really hope that this would change. I'm positive that it will. You know, if we all talk about the science, if we tackle the PCR, this is where we can undo this whole thing. So I implore everyone to study this PCR. I'm going to make beautiful materials available, you know, that you can share with other people so they can do these searches we've done, and then listen to people, let them debunk it, right? Say that what I've done here is wrong and point out why it is wrong. You know, these are the things I would want to absolutely, I want someone to challenge me. Okay. All right, everyone. Well, thank you very much. And I hope to see you. I will be doing webinars pretty, you know, fairly regularly now. I think I'm going to do like educational webinars. Then I would do webinars that are focused on healthcare, you know, because with what's going on now, you can see that healthcare it's just falling to pieces because it's all COVID. So we are all being denied healthcare. The more reason why, probably for the better, the more reason why we should take, you know, our healthcare into our own hands. And the human body is amazing. So I'm going to be doing specific webinars on health problems, how we can improve our health, right? So we all stay healthy, and then continue to do the best we can to expose this evil, because that's what it is. All right. Thank you. Thank you, everyone. Thank you very much, and see you sometime soon. Thank you.